All right, so our next contestant is Jennifer Chlebek. She is from the Physical Life Sciences Directorate, and the title of her slam is Genetically Engineered Microorganisms, a Novel Solution, or the Next Biothreat.
Before humans even knew what bacteria were, we've been using them to make some of the best foods that we have, like cheese, bread, yogurt, and beer. Today, we now have the tools to modify the DNA of bacteria to give them new capabilities, like producing medicines for humans, delivering fertilizers to crops, or cleaning up oil spills in the oceans. All of these are good ideas, but the release of genetically modified organisms, or GMOs, into the environment could become a biological threat instead of being beneficial. In my work, I'm developing strategies to prevent the next Jurassic Park of bacteria from happening. Let me give you an example. Let's say a biotech company modified the DNA of bacteria to produce a fertilizer. And then a farmer took this GMO, mixed it with the soil, and bam, they would get healthier, higher yielding crops. Now, a problem here is that these GMO bacteria are actually not the only ones in the soil. There are also native soil bacteria around as well. One biothreat is that our modified DNA could transfer from our GMO into the native soil bacteria. This is bad because now the native bacteria with the new DNA could produce too much fertilizer, which could harm our farmers' crops. So if we want to use GMO bacteria, how do we do it safely? To do that, I'm using a process called genetic entanglement. Using this novel technique, I can design DNA to make not only one, but two separate molecules. So I took my green DNA and I entangled it with a toxin so that this single strand of DNA produces both the green fertilizer and the pink toxin. The idea here is that now if the modified DNA transferred into the soil bacteria, that they would produce this toxin and die, which would protect the natural ecosystem. Next, I tested to see if this actually works. So what I did was to measure the transfer of the DNA from our GMO into many different soil bacteria. You can see in the graph on the left that when I measured this DNA without the toxin, I see a high level of transfer. However, if I entangle the DNA with a toxin, I see that this transfer is dramatically reduced. This suggests we can use genetic entanglement to decrease environmental contamination 100,000-fold. This not only helps our farmer, it brings us one step closer to widely using GMO bacteria for human benefit safely.